cycle. Previously, you learned about cell communications. Now we're going to take a look at how some of those communications manifest themselves in the cell cycle and its controls. By the end of this lecture, you should be able to describe the main events of each phase of the cell cycle, as well as have an understanding of its checkpoints and illustrate how those work. In addition, you'll have some idea of how failure of these checkpoints failure of these cell cycle controls could result in uncontrolled cell division or cancers. So let's begin by looking at the cell cycle itself. You're probably familiar with mitosis and interphase. Interphase is the green portion of the cell cycle, and mitosis and cytokinesis are in the blue portion. Mitosis and cytokinesis are a relatively short part of each cell's life. It spends most of its time in other portions of the cycle, or interphase. So mitosis, cytokinesis, right after mitosis and cytokinesis, the cell is half the size that it used to be, and so it needs to grow to its regular size. So gap 1 phase, or growth phase 1, is about cell growth back to its normal size. Now, if a cell is fated to divide again, then we might move forward in the cycle. However, most cells don't divide and then divide immediately again. They'll spend quite some time in the gap 1 phase. For example, if we were looking at muscle tissue or neurons in the brain or fat cells, they're really not going to divide again. And so when a cell is not going to divide again, we might call it G0 even, because they're not going to move all the way through the gap 1 phase. Towards the end of the gap 1 phase, we're beginning preparations for synthesis of DNA, getting ready to duplicate those chromosomes so that we have 2 copies in order to move into cell division and separate those copies into other cells. So in S phase, or synthesis phase, is where we have DNA replication occur. We're going to cover the details of DNA replication in a future lecture. But for now, once replication has occurred, we have 2 copies of each chromosome, we move into the G2 phase. And in G2, all the mechanisms that need to come into place for cell division are being produced by the cell. So G2 is completed, and the cell can then move into mitosis and cytokinesis, and the cycle continues with a pause, perhaps, in G1 phase. So let's look now at some of the details of each of these phases in the cell cycle and how they're controlled. What really determines whether a cell moves on through each of these phases has to do with some of the cell signaling and communication that we've previously learned about. So to begin with, we have 3 checkpoints, 3 major checkpoints. There's a checkpoint between G1 and S. We need to make sure everything's ready for synthesis to occur, otherwise we don't need to move into synthesis yet. So when the cell decides to divide, it's really getting signals about whether it's ready to divide. Some of these might be external signals. For example, I introduced previously autocrine signaling. During development, a cell is going to produce some substances that say, hey, I'm ready to move into another round of cell division. In this case, they spend a short amount of time in G1 and move straight on into synthesis and continue division. So growth factors are involved. You'll recall that growth factors often work with receptor tyrosine kinases. This is one of those checkpoints that once a cell has put together the machinery for DNA synthesis, we're not going to go back, right? So DNA synthesis enzymes are in place. We're moving forward into the synthesis phase. So the next checkpoint is the G2M checkpoint. So a checkpoint to see that everything is ready, all the machinery is actually in place to go into mitosis or cell division. We need to have proteins, microtubules, so we can produce the spindle apparatus that's involved in cell division, and so on and so forth. If we are ready for that, then we move into mitosis. And so we have M-phase promoting factors that help a cell decide if it's ready to move into M-phase. And these mitosis promoting factors are going to assess whether the DNA was replicated correctly, and if it was replicated correctly, can we move on into M-phase? Because we don't want to have improperly replicated DNA going into M-phase and having cell division. So we have natural checks and controls to make sure everything is in place and we're good to go. The final checkpoint that we'll look at here is the spindle checkpoint. Now the spindle checkpoint is where we're checking that all the machinery, once we've lined up chromosomes on the metaphase plate, which we'll learn about when we explore M-phase, once the chromosomes are lined up on the metaphase plate and the fibers are in place to pull the chromosomes apart, we want to check that they're actually attached properly and that we're going to separate chromosomes in a manner that ends up having one of each copy in each cell. If things are not set up properly, then we won't proceed. Again, checks and balances are in place to make sure that we're ready to separate the chromosomes and actually divide the nucleus and then go through cytokinesis and divide the cell. So what happens here is if it's not ready to go, then the cell does not divide. Sometimes the cell will be destroyed. Sometimes the DNA will be repaired before we go into cell division, but it's another irreversible checkpoint. Once you pass it, cell division is happening. So chromosomes are going to be arranged correctly or incorrectly. If they're not arranged correctly, no moving forward. And if the spindle fibers are not attached correctly to the kinetochores in order to pull the chromosomes apart, then we're not moving forward. So what are these mitosis promoting factors, though? It turns out that there are two proteins involved in moving forward into mitosis and initiating the transcription and translation of all of the proteins, microtubules and such, that are involved in pulling chromosomes apart or breaking the bonds between chromosomes. We have cyclin-dependent kinases, or CDKs, as well as cyclins. The CDKs are dependent on the cyclins. There are a number of different cyclins and CDKs that we've identified, and different ones work at different checkpoints, which makes sense because if the same ones worked, then we would promote one phase, which would also promote the other. So we have to have different partnerships of cyclin-dependent kinases and cyclins. So the cyclin-dependent kinases are activated by cyclin. We have a standard level of the cyclin-dependent kinases in the cell. Of course, a few more are made when it's time to go into mitosis. As we prepare and things are getting ready to go through one of the checkpoints, the cyclin content will increase, and we'll see more and more cyclins, and we'll see phosphorylation so that the cyclins can bind onto the cyclin-dependent kinases and become phosphorylated and start the process of the next phase of the cell cycle. So the first checkpoint that we'll look at in detail is this G1S checkpoint. The G1S checkpoint involves cyclin-dependent kinase 2, probably because it was the second cyclin-dependent kinase that we learned about. Cyclin-dependent kinases in the cell at a fairly consistent level, and then increasing levels of cyclin-dependent kinase, or cyclin E, are produced. As we see higher and higher levels of cyclin E, we see more cyclin E bound to the cyclin-dependent kinase for that step in the game. And that will see activation so that we're producing all of the factors that are necessary to move into S phase. Everything's checked, everything's balanced, we're good to go. Increasing the enzyme production for DNA synthesis is going to happen after this checkpoint, so it's irreversible. We are going to be synthesizing DNA once it's passed. The next checkpoint is the G2M checkpoint, and at this checkpoint we have a different cyclin. Makes sense to have a different one, right? Otherwise the cyclin-dependent kinase and the cyclin from the previous phase could stimulate the cell to move forward. We don't want that. So a different cyclin and a different cyclin-dependent kinase come together in order to promote movement into